Hello, is my voice audible? Is my voice audible to you? Is my voice audible to all of you? Okay, good. Okay, so then let's start with the today's presentation. Today's presentation is on polymerase chain reaction. Okay, so let me begin. Welcome all of you students. Welcome all of you. Welcome to the today's lecture. So today we are going to start with the polymerase chain reaction, that is PCR. Okay. So let's start with the PCR first. I think all of you are able to see the screen here. Uh, okay. So if you are not able to see the screen clearly, please keep your resolution of YouTube to the maximum so that you can see it. Okay, so let's start with the PCR. PCR is the polymerase chain reaction. Simply, the full form of PCR is polymerase chain reaction. Okay, so let's see what are the different constituents that are present in PCR. So PCR is generally used to amplify the DNA. Okay, so it is just like a Xerox machine in which you add certain copies of DNA and you amplify the number of copies, or you make the number of copies. Okay, so that is the primary role of PCR. Let's see some history about PCR. So PCR was first time invented in 1983 by Dr. Carey Mullis, and the first publication of the PCR was done by Citus Corporation that appeared in Science Journal. Okay, so what is the PCR using? So it uses the polymer purified Taq polymerase, which was first time done in 1986, and then Perkin Elmer introduced the first automated PCR thermocycler. Okay, so indeed a PCR was very important contribution or very important step in the science in which we were able to amplify the DNA without a live system. Okay, so I think here the DNA amplification is done. In DNA amplification, for example, in crime scene, you have a very small amount of DNA. Okay, those sample which is present is very small. Or if you have to take another example, is for archaeological evidences or fossil fuels, where the amount of sample is very small, where you require more amount of DNA for the analysis. Yes. So since the samples is available is very small, so the DNA sample is not available in ample amount. Okay. So here this PCR comes into play, where we can amplify the amount of DNA even if we are having a very small amount of sample. Okay, or if you have to study a part of a gene, then you have to do the sequencing of the gene, and after sequencing we have to amplify that particular fragment. Okay, so DNA amplification is very important in such cases. So earlier, when the PCR was not invented, the scientists were amplifying their fragment by cloning that in the different cells. Okay, you can see here there are different cells that are available in which the cloning was done. Okay, so scientists used to clone the DNA using this. Scientists used to clone the DNA using the bacteria. They used to add the fragment into the bacterial cell, and then they used to amplify. Okay, so here actually the amplification you can see here. Okay, so the amplification was done using bacteria. Okay, but gradually this used to take a longer time. Okay, but when this was PCR was invented, actually, then the time was cut short. Within 1.5 hours you can get millions of copies of DNA, and in cloning it used to take a larger time. Okay, so PCR was indeed a very useful technique. Okay, so what does the PCR means? It means to amplify a particular piece of DNA, okay, in a very short time. Okay, you can make billions of copies of DNA in a very short time. So actually it mimics the DNA replication. It is the laboratory version of DNA replication in a cell, where whatever the machinery required for replication, that machinery you have isolated in a test tube. So it is an in vitro replication also. Okay, so which just like in vivo replication happens in test tube. Okay, so this PCR is nothing but a in vitro replication that we are doing in a test tube. Okay, so it is an in vitro technique of amplification of region of a DNA that lies between two regions of known sequence. So what is the important thing here? You should know the sequence of the two ends, okay, so that you can amplify the DNA. Okay, so there is only one, that is criteria, that you should know the sequence of the sequence prior to the gene to be amplified and after the gene to be amplified. Okay, these two sequences you should know. Okay, so these two sequences will be called as forward and reverse primer. You know the DNA is having the polarity. You can see here the upper strand is 5' to 3', and the lower strand is 5' to 3' on the opposite end. Okay, so here therefore you require two different sequences on this side and this side. Okay, there are the two different sequences that is required. Okay, the one called forward primer, which has the same sequence as the forward DNA, and this is complementary to the upper strand. You can see here the forward strand and the reverse primer is attached here. The reverse strand forward primer is attached here. Okay, so there are two primers, forward primer and reverse primer. These two primers is attached to get a amplification here. Okay, so if you are using only one primer, then only one strand of the DNA will be amplified. Okay, so you have to use both the primers for getting a double stranded DNA amplification. Okay, so let's see the components of PCR. In PCR you require, as I told you, you require a in vitro assembly. So you require nucleotides in the form of DNTPs, that is deoxynucleotide triphosphate. Okay, then you require primer, that is two sets, forward primer and reverse primer. Okay, then DNA sample is also required, which you want to amplify. Obviously, tagged DNA polymerase is very important here. Tagged DNA polymerase is the DNA polymerase obtained from Thermus aquaticus. This is an thermophilic bacteria that grows in a hot springs on an area where temperature is very hot, close to 100 degrees Celsius, or geothermal vents. Okay, so in that case, in that organism, this thermostable polymerase is there, and this is isolated. Why? Because in case of PCR, the temperature is ranging or varying. Okay, so to change the varying temperature or to cope up with the varying temperature, this enzyme is used. If you are using a regular enzyme, then what is going to happen? The enzyme is going to denature. Okay, so therefore thermostable enzyme from tagged DNA polymerase is used. There is a problem with this DNA polymerase, that it is polymerizing at a very high rate, but it is not having a proofreading activity. Okay, so sometimes what we do, we mix it with other polymerases, other thermostable polymerases, such as PFU polymerase, which also does the proofreading. Okay, so along with this, you mix buffer here. So buffer is PCR buffer, in which all these components are mixed, and then obviously you have to mix this all in PCR tubes. Okay, and then one more factor is also required here. This is MgCl2. Now, why MgCl2 is important? See, MgCl2, Mg is the cofactor for DNA polymerase enzyme here, that is the tagged DNA polymerase. So it has to be added in the reaction mixture. Okay, so these are the different components. Additional reagents may be added depending on the use or depending on the need. Okay, so this is the overall PCR reaction. This happens in around 3 steps: denaturation, annealing, and extension. There are different temperatures for annealing and all. So let's see each step in details. Okay, the first step is denaturation. Okay, the double-stranded DNA is denatured by heating, and the heating is typically done at 95 degrees Celsius. Okay, to separate the double-stranded DNA. Now, why to separate the double-stranded DNA? Remember, replication will not happen until and unless the DNA is separated. In regular replication also, DNA helicase enzyme separates the two strands. Here, we are separating in the two strands by using the heat. Okay, we have to break the bonds here, breaking dash bonds. So we have to break the hydrogen bond between the two strands. Okay, then only the two strand is going to separate. Okay, now you should know that the strand is having certain polarity. Okay, it is having certain direction. Okay, So the denaturation is always occurring at 94 to 97 degrees Celsius. Normally, when we teach or when we teach about the PCR, we teach only three steps, that is denaturation, annealing, and extension. But one more step is there prior to denaturation, that is initial denaturation. Remember, at the starting, the DNA is totally in the double stranded form. Okay, so it has to be made in single stranded form. So, a additional step, that is initial denaturation, is done, and this is done at starting only, and last step is final denaturation, that is also done after the final extension is over. Okay, so let's see the three steps first. This is the denaturation, and after the denaturation, then the primers that are present in the reaction mixture, that is forward, this is the forward, and other one is the reverse primer, that is added into the reaction mixture. Okay, now when the primer is added here, it is going to anneal at this position. Okay, since the primer is oligomeric primer, it is about 18 base 15 to 18 base primer is there, and it adds to the or goes and attaches to the complementary region that is present at the starting and the ending of the fragment that we want to amplify. Okay, so primer anneals at a particular temperature. Okay, so the temperature has to be brought down to 50 to 65 degrees Celsius. It may be different for different reactions, so the temperature should be brought down to 50 to 60. Now, see, the initial denaturation or denaturation step occurs for around 2 minutes. Okay, and immediately the temperature has to be reduced to such a low temperature, that is 50 to 65. So this can happen in thermocycler only. Okay, so we can see that we will see the thermocycler also in subsequent slides. Okay, so after annealing step, what will happen? The DNA polymerase or the tag DNA polymerase is going to attach to the both positions, and it will extend the DNA in the 3' direction. Now the extension temperature is 72 degrees Celsius. So extension happens at 72 degrees Celsius. That is the optimum temperature for tag polymerase activity. Okay, so tag polymerase synthesizes the double-stranded DNA, and now the double-stranded DNA is prepared. Okay. Again, the step will repeat. Again, there will be denaturation. The two DNA strands, double-stranded DNA strands that are being formed, are again separated, and then again annealing will take place. And this will keep on repeating till the number of cycles you want. Okay. So it will be exponential amplification. If you have to set the PCR reaction, this is the way how you can set the PCR reaction. First of all, first cycle is denaturation. I already told you in general, in practical, before denaturation step, there is an initial denaturation step. Okay, and then after the extension, there is a final extension step. Okay, that is always done at the starting and at the last. Okay, and in the middle, denaturation, annealing, and extension, this cycle is repeated for about 30 to 35 times, or even 45 times also in some cases. Okay, so this is the cycle that is repeated, and after this much cycles happens, we can get the millions and billions of copies of DNA. Okay, so this is how this happens. The red colored is the wanted gene here. Okay, we put primers on both sides. In first cycle, this DNA is denatured and opened up, and two separate DNA double-stranded strands are opened. So for 2 raised to 2 cycles, you get 4 copies of DNA. Okay, and then for 2 raised to 3 cycles, you get 8 copies, then 16, and likewise for about 2 raised to 38 cycles, or 2 raised to 38 cycles, you get about 68 billion copies. Okay, so it amplifies exponentially. You can see the number of copies you are getting is amplifying exponentially. Okay, so these many number of copies you are getting after the PCR is over. Okay, so when you do the Agarose Gel Electrophoresis of this kind of PCR, this is the marker in the first lane. This is the regular band, and you can see the target sequence that is being amplified here, and this is how you are going to get the PCR product. Okay, you can purify also this product from the gel by silica gel extraction method. Okay, so the important thing here is the primer designing. Okay, how to design the primer. Okay, normally the DNA is present in any organism. The important thing is primer designing. Okay, for that you should know the sequence prior to the gene that you want to amplify, and after the gene that you want to amplify. So if you know the around 30 to 50 base pair sequence, then you can easily prepare the primer. Okay, so primer preparation is very important, because primer is going to bind at the starting and ending point of the gene, that is as a forward sequence or forward primer and reverse primer. So primer sequence is very important. Okay, what the sequences the primer is having. About 40 to 60% GC content in the primer is we want 40 to 60% of GC content in primer, okay, so that it can bind firmly. Then primer length is also very important. It should be neither too short or not too lengthy, because optimum primer length is from about 18 to 20 base pairs, okay. So you should get the primer length there only. As I told you, GC content is also important. 40 to 60% GC content should be there. GC clamping should also be there, okay. So the optimum amount of GC content plus repetitive sequence also must not be there, which will form a clamp or a hairpin structure. That should not be there, okay. And then melting temperature should be also optimum. What is the melting temperature? It is the temperature at which half of the DNA is melted, okay. So therefore the GC content is required so that the primer will not melt easily, and it will sustain the temperature, okay. Then annealing temperature should also be optimum for the primers, so that it can anneal easily to the DNA strands, okay. How to select the primer? See, primer, what is a primer? It's an oligonucleotide sequence and will target the specific DNA and it will bind at that specific sequence, okay. So you should know that finding, so what is the optimum length? I'll tell you how to choose the optimum length, okay. So there are 4 bases A, G, C, T in the DNA, okay. So for every probability of every base is about 1/4. Yes, probability of finding a single base is about 1/4. So probability of finding a dinucleotide or 2 base pairs together out of this A, G, C, T is 1 by 16, obviously, and finding a 4 base sequence together is 1 upon 256. If you do the probability here, the finding the base of 4 nucleotide sequence together is 1 upon 256, okay. So thus a 16 base sequence will statistically be present only once in every 416 bases, okay. So when you take a 16 base sequence or 16 bases together in a primer, so the probability of this getting this base is once in every 416 bases. So if you multiply it with the number of bases, you get around 4 billion bases. That is about the size of a human genome or a maize genome. It is far greater than E. coli genome, okay. So therefore, this is a uniqueness of the primer. Therefore, primer synthesis is very important, or selection of primer is very important. Primer specificity is also very important. Okay, so primer should bind at the required sequence only. If it is not binding at the required sequence, then what will happen? It will not give the sequence that we want to amplify. Okay, forward and reverse primer already I have discussed with you. And then DNA polymerase is very important here. I already told you that this is being isolated from a geothermal vent that is found in Yellowstone National Park in United States. And this organism is named Thermus aquaticus. There are other polymerases also, such as Pyrococcus furiosus, PFU polymerase. This is another polymerase that survives the high temperature. Okay, so both of them can be used in the PCR. Okay. Then for primer preparation, obviously you have to cut the primer sequence from certain DNA, and RFLP, that is random restriction fragment length polymorphism, comes into play. You can use the restriction enzyme to cut the DNA and form a primer. Now this primer can be used for PCR. Okay. So based on the type of amplification you are doing or a desirable trait in the amplification, the PCR may be of different types. There are around 36 number of PCRs. Okay, so you can see AFLP PCR, allele specific PCR, ALU PCR, assembly PCR, asymmetric PCR, cold PCR, colony PCR is one of the important one. Then conventional PCR is the regular PCR that we do. Then digital, fast cycling, high fidelity, hot start PCR, in situ PCR, inter-sequence specific PCR, then inverse PCR, late PCR, ligation mediated PCR that is used for sequencing also, ligation mediated PCR, then long range PCR, multiplex PCR, nanoparticle assisted PCR, fluorescent PCR, real time PCR is one of the important. Then RT-PCR, that is reverse transcriptase PCR. It is very important, and it is used. It was used widely in the COVID pandemic for the diagnosis of COVID. Okay, then such kind of so many number of PCRs are there depending on the different applications of PCR. Okay, so indeed the PCR is very important in today's world. Okay, so now let's see the what is thermocycler. Now remember, I have told you or explained you the whole process of PCR now. Okay. Now you know the PCR. Now it has to be done in a certain machine. Okay. So that machine is called thermocycler. Thermocycler. It is a PCR machine or DNA amplifier. Is a laboratory apparatus most commonly used to amplify the segments of DNA via polymerase chain reaction. So you are using a PCR or polymerase chain reaction in a thermocycler to amplify the DNA. Okay. So what this device consists of, it device have a thermal block. At the center, there is a presence of thermal block with lots of holes in it. You put the PCR tube in it here. Remember the PCR tubes has to be, autoclaved and it should be nucleus free. Okay. Then this reaction mixture is prepared and then inserted into this. And you set a proper timer or proper protocol according to which the PCR is going to happen. For example, denaturation, initial denaturation will be for 2 minutes, then denaturation step will be for 1 minute, annealing for 2 minutes, then final extension for 2 minutes, and final denaturation, final extension for about 5 minutes. Okay, so like that you have to design the PCR protocol according to the need. Okay, and then you have to set the PCR, and you have to put the program and put the tubes and start the PCR. Okay, so if you want to see how to set a basic PCR, there is a video available in my channel. You can refer to that video, how to set a basic thermocycler. Okay, so these are all the steps where you have to set a thermocycler and then you can start the PCR reaction. I will show you certain photos of thermocycler. You can see this was a very early PCR machine in which robotic arm used to add the enzyme every time, because the thermos aquaticus or the tag DNA polymerase was not discovered till then. Okay, so after every cycle the DNA polymerase would denature and new polymerase has to be added, and the robotic arm used to add the new polymerase after every cycle. Okay, so this was one of the ancient PCR in about close to 1986 or 1985. Okay, this is another prototype of a automated PCR machine, and this is one of the modern PCR which works very fast. This you can see is the open lid of a particular PCR machine, or the thermal block is opened here. You can see the number of holes. This element present here is very important because it immediately reduces or increases the temperature. So now the flexibility of increasing and decreasing the temperature is very important in this case. So therefore that is called as a thermal block. You can see the PCR tubes are kept on that. Okay, so this is very important, and therefore the lid is closed and then PCR is performed. Okay, so indeed the PCR is very important for applications such as genetic engineering and in biotechnology and in disease diagnosis also. Okay, so this was all about the PCR and its types. You can see I am showing you another PCR. This is the basic PCR, that is a thermocycler. Then these are the few other examples also. Okay, today in this lecture I told you about different polymerase chain reaction and thermocyclers and its types. I have shown you certain images of thermocycler also. I hope you have understood the whole mechanism of PCR and thermocycler. So thank you for joining today's lecture. Thank you, students.